So, to defeating, we require the knowledge that we now have in these subjects, and requires, we've heard about selecting for extended longevity, which is doable, except it takes a long time in vertebrates, and genetic modifications that extend longevity. Oh, one more thing here. Science versus engineering. If you look at most of the funding for aging right now, it's going into science, why we age. Most of it is not going into make me a solution for aging. And as a result, we're ending up with lots more information about what we understand about aging, but very little information about what we have to build to solve it. But these mice, the clock knockout mice or half knockout mice, live longer. Now, this knocks out coenzyme Q production, which is interesting enough in and of itself because, whoa, everyone's saying take more enzyme Q, but less enzyme Q is better. Okay. The MCAT mice are catalase targeted to the mitochondria. Those seem to have an extended lifespan. These mice here increase telomere expression, or telomerase expression, but they've also got a better P53, extra copies of P53 and P16 and P19, and that basically says these are cancer resistant and therefore they live longer. Now, this is something we've discussed. You want to increase the length regulation in the future. You would like to have the telomere length be maintained intact in all cells that are not about to become cancerous. So you can turn telomerase on in cells that do not have a stress response going on. But then otherwise it should be shut down. Then this is the recent discovery that mole rats seem to not get cancer, and that's because they have an additional restriction involving this gene that prevents the cells from replicating when they come into contact with each other, which might be useful. The mitochondrial membrane makeup, birds and bees seem to be able to pull off their tricks because they have a better mitochondria. General perspective is copy and steal. Information is redundant in these genomes. This is important because this is the only species I've found, plus the other radiation-resistant species, of which there are 4, that have a different double-strand break DNA repair pathway. It has redundancy in the number of genome copies, and it has a very unique double-strand break repair process. Now, life on hold is hydrogen sulfide, has recently been discovered to be able to decrease your temperature and decrease your metabolic state, therefore decreasing the production of free radicals. And they're going with, Icaria is the company, and it's going after being able to put people in suspended animation, essentially. Those are the genes that have, some of the organisms have currently been sequenced. And the comparative genomics between these allows you to see that the double-strand break repair response is identical over hundreds of millions of years. And that's the roadmap for lifespan extension. So this is what we have now. This is what we have now too. You can go to 23andMe and find out your risk for various diseases. This we have now in transplants. The problem is the shortage of organs. This we have now. There are 2,600 stem cell therapy trials going on in the US today. And the problem is you don't have pristine stem cells. All of these cell therapies are being done with either cells from young individuals or cells in bulk, where the quality of the genomes of a trillion cells is subject to suspicion. Did that go back? No, that's okay. And Sangamo is a company in San Francisco that has discovered got a good way of replacing single genes, so you can correct defects up here, but it currently is only being targeted towards being able to take cells out of the body, correct the gene, and put the cells back in the body. Then you've got the suspended animation, which I just discussed. If you've got people that are going to die, better to put them in suspended animation than let them die. That is somewhat better than cryonics because you don't have to deal with the damage when you're trying to bring them back. You ultimately want to augment the genome, and then you want to get nanotechnology on board. Pristine stem cells are basically what I've been working on the last couple of years. They're the idea that you can affordably select cells that are pristine. All the mutations are in the junk DNA and allow you to get cells like when you were born, which are the cells that you would like to grow organs from. And they can actually be augmented. And so you would like to be able to produce chromosomes that you can add that fix the defects, that produce free radicals, that kind of thing. So genome reengineering is where we'd like to get to. You'd like to be able to add a chromosome to the stem cells that cover all of these things. Some things are easy, so as upregulating or making better antioxidant defenses, upregulating the proteasome function, because the genes for these are already there. All you have to do is turn them on more. Some of these are very hard, such as removing toxic metals, to remove iron and copper, which are essential metals in the enzymatic system. There are about 400 genes that use iron and maybe slightly less than 200 that use copper, proteins that use these metals. And so removing these means you're really doing major surgery on the functioning of the genome. You want fault tolerance, so better clotting, a variety of things like that. You would like cells to replicate faster. You ultimately want to get so you can replace the mitochondrial chain and correct the mutations that were put into it from the get-go, which allow the generation of free radicals. That's a really radical change. That's changing the fundamental architecture of cells. And then you would like to be able to extend these. If you're going to add a chromosome, add a chromosome so that you can hibernate. So when the weather's bad some year, you say, I'll just go to sleep and wake up next year. Shells would be nice, means you can't be crushed by buildings, et cetera. So there's all kinds of things you can imagine once you get started down this path.
So Robert, you have 2 minutes.
Okay. But these are the things which ultimately limit your longevity. Once you've created a non-aging genome, or a genome that ages over thousands of years, then you've got to deal with fires, earthquakes, terrorist attacks. The idea of having a conference every year in LA, in my mind, is a really scary idea. You've also got these weapons potentially to worry about. You've got an evolving artificial intelligence that doesn't have good controls on it. These are the things, 65 million years ago, a lot of species got wiped out. The mammals got wiped out. All the large species that were land, surface-dwelling animals got wiped out because of the firestorm that resulted from the impact. Anything that was above ground, not underwater, and not in a burrow, basically became extinct at that time. And then you've also got supernovas, which don't occur frequently or close enough to us, but ultimately these are things you have to worry about. And these are the solutions that you can engineer. Ultimately, you get down here, which is mind uploading and backup copies, which are radical concepts most people do not like to grasp. They say, I'm me, I'm this body. No, you're not you, this body. From an information science standpoint, you are you, the memories that are kept up here. And you can you can die. And to me, someone is not dead until their brain has completely disappeared. So you could slice and scan it and recreate the person in a computer. Or you've got nanorobot-enabled offloading, which means you offload before you die, and you've got copies. At any rate, that's what it ends up looking like. That, for the people who don't know, is called a matryoshka brain. And a matryoshka brain potentially has the lifespan because these little satellites here orbiting the star all contain uploaded individuals. And the lifetime of one of these is the lifetime of the star, which in our case means a couple of more billion years. If you do advanced stellar engineering, you can extend the life of a star out to about 100 billion years. And that is the outside limit, probably, unless you move your star to another star and start. No, I mean the star. You can move the star. Because where all this material comes from here, this is potentially about twice the diameter of the solar system at the far end, and then the inside is like inside of the orbit of Mercury. And if you move yourself one way or another to another star, you can start all over again. So you get another 10 billion years, then you use a star that will last another 100 billion years. So then, as Ralph commented the other day, What your real worry is the heat death of the universe. So.
And on that cheerful note, we'll have questions. Thank you, Robert.